Today the class is on lipid metabolism. So, a short intro about lipids. So, lipids are heterogeneous group of water, insoluble organic molecules that can be extracted from tissues by non-polar solvents. It is a major source of energy for the body. Also provides the hydrophobic barriers that permits partitioning of aqueous contents of the cells and subcellular structures. Fat-soluble vitamins have regulatory or coenzyme role in the body. Prostaglandins and steroid hormones, they have a major role in controlling the body's homeostasis. It gives shape and contour to the body and protects the internal organs by providing a cushioning effect. These are the various functions of lipids. Dietary fat composition. More than 95% are triglycerides and others are cholesterol, cholesterol esters, phospholipids, and unesterified fatty acids. In animal sources, dairy products like meat, butter, and ghee, meat and fish, pork, and eggs. These are the animal sources which has lipids. Vegetable sources are cooking oils, sunflower oil, mustard oil, groundnut oil, fats from other vegetable sources. How the digestion is going on. So in the mouth, how the digestion is taking place. Hydrolysis of triglycerols or triglyceride is initiated by the lingual and the gastric lipases, which attacks the ester bond of forming 1 to 2 diacylglycerols and free fatty acids, which aids in emulsification. What is the role of lingual lipase? This is secreted by dorsal surface of tongue, active at low pH, that is from 2.0 to 7.5. Optimum pH is 4. That enzymes pH is 4.0 to 4.5. Ideal substrate is short chain triglycerides. Milk fat contains short chain fatty acids, which are esterified at the 3rd position. Thus, it is the best substrate for lingual lipase. Enzymatic action continues in stomach. Short chain fatty acids, they are released and they are absorbed directly from the stomach wall and enters the portal way. So let's have a look at this, how the triglyceride is degraded. Degradation of triglyceride. So fat molecule, water, and acted upon by a digestive enzyme, mainly on triglyceride, and it is cleaved into 3 parts, that is fatty acids and glycerol. So triglycerides are degraded by lipases to form 3 fatty acids and glycerol. What happens in the stomach? How the digestion is going on in stomach? So here the enzyme is gastric lipase, secreted in small amounts, more effective at alkaline pH. Average pH is 7.8. Requires the presence of calcium. Less effective in stomach due to acidic pH when intestinal contents are regurgitated into the gastric lumen. Not effective for long chain fatty acids. Most effective for short and medium chain fatty acid. Milk, egg yolk, and fats containing short chain fatty acids are suitable substitutes for its action. So this picture will help you to understand how it is being uh, helps in digestion in the stomach. Role of fats in gastric emptying. Fats, they delay the rate of emptying of stomach. Action is brought about by secretion of enterogastrone. Enterogastrone inhibits gastric motility and this retards the discharge of bolus of food from the stomach. Thus, fats have a high satiety value. Significance of lingual and gastric lipases. They play important role in lipid digestion in humans. Since milk is the only source of energy, important digestive enzymes in pancreatic insufficiency, such as cystic fibrosis or other pancreatic disorders. Lingual and gastric lipases, they can degrade diglycerides with short and medium chain fatty acids in patients with pancreatic disorders despite a near or complete absence of pancreatic lipase. Emulsification and digestion. So these are the two processes which are taking place during digestion. So lipids are hydrophobic and thus they are poorly soluble in the aqueous environment of the digestive tract. The digestive enzyme, the lipase, which is water soluble, and it can only work at the surface of fat globules. Digestion is greatly aided by emulsification, which is nothing but the breaking of fat globules into much smaller emulsion droplets. Emulsification and digestion, which are continuing. So here, the triacyl glycerol digestion occurs at the lipid-water interfaces. The rate of triglyceride digestion depends on the surface area of this interface, which is increased by churning peristaltic movements of the intestine. This combined with the emulsifying action of bile salts, the critical process of emulsification takes place in the duodenum. How digestion takes place in small intestine. It is the major site of fat digestion. The effective digestion due to the presence of pancreatic lipase and bile salts. Bile salts, they act as effective emulsifying agents for fats. Secretion of pancreatic juice is stimulated by passage of acid gastric contents into the duodenum by secretion of secretin, cholecystokinin, and pancreozymin, the gastrointestinal hormones. Gastrointestinal hormones are secretin, which increases the secretion of electrolytes and fluid components of the pancreatic juice. Pancreozymin of CCK, PZ, which stimulates the secretion of the pancreatic enzymes. So cholecystokinin of CCK, PZ, which causes the contraction of the gallbladder and discharges the bile into the duodenum. Next one is hepatocrinin. This is released by intestinal mucosa and stimulates more bile formation, which is relatively poor in bile acid content. The contents of pancreatic juice are pancreatic lipase, which helps in digestion of triglycerides. Phospholipase A2 is for digestion of phospholipids. Cholesterol esterase for the digestion of cholesterol esters. The use of bile salts in digestion. Bile salts are required for the proper functioning of the pancreatic lipase enzyme. Bile salts help in combination of lipase with two molecules of small protein called as colipase. This combination enhances the lipase activity. Bile salts also help in the emulsification of. So when you see this picture, you have a big triglyceride particle or a molecule, and where you have lipase, the enzyme, and a small co-lipase molecules. So this will act on the triglyceride. Bile salts. Bile salts are synthesized in the liver and stored in the gallbladder. They are derivatives of cholesterol. They consist of a spiral ring structure with a side chain to which a molecule of glycine or taurine is covalently attached by an amide linkage. So you can see the picture here, how it is attached. Bile salts are formed from bile acids. The primary bile acids are folic acid, found in the large amount, and chenodeoxycholic acid. These are the two primary bile acids. The primary bile acids, they enter the bile as glycine or taurine conjugates. In the alkaline bile, the bile acids and their conjugates are assumed to be in the salt form, hence the term bile salts. How bile salts are synthesized? So the basic product is, or the prime product is from cholesterol. So cholesterol, it is converted to chenodeoxycholic acid. Then chenodeoxycholic acid is converted to cholic acid. And here in chenodeoxycholic acid, it is conjugated with glycine or taurine. So here, and cholic acid also same. So here it will be glycokenodeoxycholic acid and taurocenodeoxycholic acid. Here it is glycocholic acid and taurocolic acid. So it is called, after conjugating with glycine or taurine, it becomes the glycokenodeoxycholic acid and taurocenodeoxycholic acid. And the cholic acid, after conjugation with the glycine or taurine, it becomes glycocholic acid and taurocolic acid. So these are the conjugates. In humans, the ratio of glycine to the taurine conjugate is normally 3:1. Bile salts. A portion of the primary bile acids in the intestine is subjected to further changes by the activity of intestinal bacteria. They include deconjugation and 7-alpha dehydroxylation, which produces secondary bile acids, deoxycholic acid and glycocholic acid. So now we will see about how enterohepatic circulation of bile salts happens. So the bile salts present in the body are not sufficient to fully process the fats in typical meal. So they need to be recycled. This is achieved by the enterohepatic circulation. Specific transporters in terminal ileum moves bile salts from the lumen of the digestive tract to the intestinal cavity. They are then transported directly to the liver via the hepatic portal vein. Hepatocytes, they take up bile salts from the blood and increase the secretion of bile salts. Bile canaliculi, small passageways that convey bile into the larger bile ducts. 95% of the bile that is released to the small intestine is recycled via the enterohepatic circulation, while the 5% of the bile salts are lost in the feces. So in liver, the cholesterol gives rise to the formation of bile acids, and from bile acids, it gives rise to bile salts. Then next, after formation of this is being stored in gallbladder, in bile, through bile. So here what happens is, in the small intestine, the bile salts and bile acids, they are acted upon by bacteria, and secondary bile acids are formed, which are excreted. And some part from the bile acids, it will be through portal blood, it will enter into liver. Emulsification by bile salts. So let us have a glance at this. Understand this fat globule and the emulsification, how it happens. See look at the fat globules. Okay, all this, and it is being clearly shown. Bile salt. So here, the fat droplets are coated with bile salts or suspended in water. Bile salts as emulsifying agents interact with the dietary lipid particles and the aqueous duodenal contents, thereby stabilizing the lipid particles as they become smaller and preventing them from coalescing. Next is triacylglycerol degradation by pancreatic lipase. Pancreatic lipase is specific for the hydrolysis of primary ester linkages, that is fatty acids present at position 1 and 3. It cannot hydrolyze the ester linkages of position 2. Digestion of triglycerides proceeds by removal of the terminal fatty acid to produce alpha beta diglyceride. The other terminal fatty acid is then removed to produce beta monoglyceride. So it is a pictorial representation how the triacylglycerol is degraded by the enzyme pancreatic lipase. So this is position number 1, how it is degraded. And pancreatic lipase is an enzyme which breaks the bonds between the glycerol and the fatty acid at positions 1 and 3. So you get a compound. Okay, the free fatty acid. But the last fatty acid is linked by the secondary ester group, hence it can not be hydrolyzed by the pancreatic lipase. Beta monoacylglycerol can be converted to alpha monoacylglycerol by isomerase enzyme and then hydrolyzed by pancreatic lipase. The primary product of hydrolysis are beta monoacylglycerol, alpha monoacylglycerol with free fatty acids and glycerol. Emulsification and digestion of triglycerides in how it happens. So here what happens is, from the stomach, it enters and from liver and gallbladder, it enters through bile duct, the micelles of bile salts and cholesterol and lecithin. So here, what happens here is emulsification. First step, emulsification of fat droplets by bile salts. And second one is, it is hydrolyzed in and emulsified fat droplets into fatty acid and monoglycerides. And the last one is dissolving of fatty acids and monoglycerides into micelles to produce mixed micelles. Significance of pancreatic lipase. The enzyme is present in high concentration in pancreas. Only very severe pancreatic deficiency such as fibrosis results in malabsorption of fats due to impaired digestion. Orlistat is an anti-obesity drug which inhibits gastric and pancreatic lipases, thereby decreasing fat digestion and absorption, resulting in weight loss. How cholesterol ester is degraded? So the dietary cholesterol is mainly present in the free, that is non-esterified form. Only 10 to 15% is present in the esterified form. Cholesterol esters are hydrolyzed by pancreatic cholesterol esterase to produce cholesterol and free fatty acid. The enzymatic activity is greatly increased in presence of bile salts. So again, let me explain how it is with a pictorial representation, how it is being degraded. So this is a picture of cholesterol ester, and the enzyme cholesterol ester hydrolase acts on cholesterol ester to give you free fatty acid and a cholesterol molecule. Phospholipids degradation. How phospholipids are degraded? The enzyme phospholipase A2, it requires bile salts for optimum activity. It removes one fatty acid from carbon 2 of the phospholipid to form lysophospholipid. The remaining fatty acid at position 1 can be removed by lysophospholipase, leaving a glyceryl phosphoryl base which can be excreted in the feces, further degraded or absorbed. So again, with a molecular structure, how it is being degraded. Phospholipase 2 is an enzyme and you get this is a phospholipid acted upon by phospholipase A2 and you get lysophospholipid. Next, we'll move on to absorption of lipids. So all this where how it is digested, how the fat molecules are digested.